Now, this discussion would be on general embryology, but I will start the discussion on a different topic which should come before the development of the embryo, that is this topic, fertilization and implantation. Okay, to start with, fertilization means the union of the ovum and the sperm. In other words, the sperm enters the ovum. That is called fertilization. And then implantation means the attachment of the fertilized ovum, which is, by the time of fertilization, it's more than one cell, several cells. We will discuss that later. Anyway, implantation means the attachment of the fertilized ovum to the endometrium of the uterus. Uterus is the womb of a female. The uterus has several layers. This is not the time to discuss about the uterus, but anyway, uterus is something like this with a thickness, and you have what is called cervix here. So this is called endometrium, this is called perimetrium, and the middle layer, which is the thicker layer, the muscle layer, is called myometrium. So implantation is the attachment of the fertilized ovum to the endometrium of the uterus. Now, our lives begin as one cell, a single cell formed by the union of the maternal and paternal germ cells. In other words, by the fertilization of the ovum from the mother with the sperm from father. So that is the beginning of our life as one cell. And if you take a somatic cell of the body, now what is somatic? Okay, these are the terms that you might not know. Now, somatic means any body cell, any body cell other than germ cells. Okay, so other than germ cells, any other cell in any of the organs or tissues, you call them somatic cells. Okay, so our somatic cells, whether we are males or females, we have 46 chromosomes. You know all that very well. 46 chromosomes arranged in 23 pairs. Arranged in 23 pairs. And these 23 pairs, they are in 22 pairs of autosomes. 22 pairs of autosomes and one pair of sex chromosomes. One pair of sex chromosomes and 22 pairs of autosomes. Because of these pairs, 23 pairs, you call it diploid. So all somatic cells or body cells, their state is called diploid. And when the germ cells are formed, we will discuss this a little bit later. When the germ cells are formed, the number of pairs gets halved. Number of pairs gets halved. In other words, the number of chromosomes, total number of chromosomes, the 46 becomes 23. Therefore, they are called haploid. Okay, diploid and haploid. And when two haploid germ cells, the sperm and the ovum, fertilize, then the cell that is produced, again, it becomes diploid. 46 chromosomes in 23 pairs. That's how it happens. Okay. Now this is, remove this for a moment. Now this is a human karyogram. Now what has been done here is you take a somatic cell. Now you know what is a somatic cell. You take a somatic cell and lyse it. Lyse it means you destroy it, destroy the cell membrane, and you take the chromosomes out. And you pair similar chromosomes together and then arrange them in order from the largest pair of chromosomes to the smallest pair of chromosomes. Okay, especially the autosomes. And then when you arrange that, you get an arrangement like this. Up to here, you get from largest to smallest the autosomes, 22 pairs of autosomes. Then the last one is the pair of sex chromosomes. Okay. Now in this particular case, as you can see here, there is no Y chromosome. It's 2 X chromosomes. Therefore, this should be a female karyogram. And the chromosomes in pairs, for instance, if you take this one one has come from the mother, other one has come from the father of this person. And therefore, and they code the genes that are there on this chromosome, they code for the same traits. You know what are traits. And therefore, this pair of chromosomes is called homologous pair of chromosomes. That's the point I wanted to raise. It's called homologous pair of chromosomes. Then the other point is that as we discussed in the previous slide, this state where you get chromosomes in pairs, 23 pairs, is called diploid state, or you can call it 2N DNA state. So all somatic cells, body cells, are in 2N DNA state. Now, when the cells divide, there are two types of divisions. The first one is mitosis, and the second one is meiosis. Mitosis and meiosis. Mitosis takes place during growth and repair of the somatic cells, or body cells. Growth and repair of somatic cells. And meiosis takes place during gametogenesis, that is formation of ova and sperms inside the ovaries and testicles. Now, if you take a female with the genotype of 44XX, a somatic cell with this genotype will produce two somatic cells with the same genotype, 44XX. If a male with 44XY produces cells with mitosis, that cell will produce two cells with 44XY. And when it comes to meiosis, on the other hand, if the female is 44XX, the ova that are produced would be 22X, half. Okay, half of the chromosomes would be there. And if a male with 44XY produces sperms would be either 22X or 22Y. So you will get a mixture of 22X and 22Y sperms. So when they meet, then this image shows you mitosis. If you can see here, there are four main steps in mitosis: prophase, metaphase, anaphase, and telophase. So there are four main steps in mitosis. And if you look here, you can see that at the beginning of mitosis, not at the beginning actually, before the mitosis starts, the usual 2N DNA state changes. The DNA replicate and gets doubled. So ultimately, you get 4N DNA state. This is just before mitosis starts. And if you look at a chromosome, now before mitosis, if a chromosome is like this, now at this 4N DNA state, so this is 2N DNA state. At 4N DNA state, if I make it 4 here, the chromosome would look like this. So there are two strands here. Now, each of these will be called a chromatid. Each of these will be called a chromatid. Okay. So that's why you have 4N DNA state. Then during the mitosis, the process of mitosis, they get arranged in the center of the cells in a plane like this. All the chromosomes get arranged, and then a spindle is formed, which is attached to the chromosome at the centromere, and the contraction of the spindle will pull them towards the two sides of the cell, in the process breaking the chromosome in the center, and one chromatid will go to that side, the other chromatid will go to this side. So ultimately, you get two cells formed here and here in this phase, last phase, the telophase. So ending up in producing two daughter cells with 2N DNA state. So the same DNA state and the chromosomal state is restored here. So this is mitosis. Then in meiosis, in contrast to mitosis, what happens is, just like in meiosis also, just before the meiosis starts, the 2N DNA state becomes 4N DNA state by replication of DNA. Then what happens is, then actually there are two phases in meiosis, unlike in mitosis. There is first meiotic division and second meiotic division. Now, at the end of first meiotic division, what has happened is that the two homologous pair of chromosomes, one that has come from the mother and the one that has come from the father, has got separated out and gone into two cells that are produced at the end of the first meiotic division. Okay, but the chromatids are still attached to each other. They are not broken out like in mitosis. But anyway, because of the homologous pair going into the two cells, the number of DNA has got halved now. So it has come down to 2 in DNA state. Then, just like in mitosis, a new spindle is formed and the two chromatids are pulled to the two sides, and two cells are produced from each cell. So ultimately, you get four daughter cells, and the DNA has become still again halved, and then you get 1 in DNA state. So this is called the haploid state. You started with diploid state, then went up to 4 in DNA state and came back to 1 in DNA state. So 4 daughter cells will be produced with 1 in DNA. So this 1 in DNA state cells will actually represent ova and sperms, germ cells. So this is what we explained. So it starts with 2 in DNA, germ cells are 1 in DNA, and when they unite, they will form a cell, a somatic cell again, which is 46 chromosomes at 2 in DNA state. So this 46 2 in DNA state is restored. Okay. Then one few things about gametogenesis. Genesis means production. Okay. So gametogenesis means formation or production of sperms and ova. So you have two terms there. For gametogenesis is the common term. So production of ova is called oogenesis, and production of sperms is called spermatogenesis. Okay, so that is clear. And this there are a few terms now. Ovum means one egg cell. It's singular. Ova means plural. Okay, some of you might not know it. Okay, and another term for ovum is oocyte. It's more technical. Okay, oocyte. So ovum and ova. Okay, so plural is ova. Then similarly, now oogonium is plural. Oogonia is written here. Oogonia is plural. Similarly, spermatogonium is singular and spermatogonia is plural. Okay, better to know these terms first. Okay, so I will use these terms freely here. Now, in a female, during oogenesis, so going by oocyte, okay, during oogenesis, what happens is the initial cell inside the ovary is called oogonium. One cell is called oogonium. Many cells are called oogonia. And these are called stem cells. Stem cells because those are the cells from which other cells originate. And that cell undergo oogenesis and forms a oocyte. So this is actually the process of meiosis, oogenesis. But there are other steps in that. It's the basic process is meiosis. But in females, it's a bit peculiar because even though the meiosis starts, it starts very early. Meiosis for the formation of ova from oogonia starts at the time when the baby is in the mother's womb, at the time of embryonic period. It starts very early, but it gets arrested in the first meiotic division. Now the meiotic means first division of meiosis. Okay, so at the end of the first meiotic division, it gets arrested, and it remains arrested until the baby is born, and it remains arrested until the child reaches puberty. So up to puberty, it is still arrested. Then at the time of puberty, when at the end of each cycle, one ovum, it's released from its arrest and it starts its meiotic division. Okay, and then still, it does not complete, even though we call it mature ovum and oocyte. This is not inside the ovary. So what is released from the ovary is still not fully completed in the form when it comes to its meiotic division. The maturation completes at the time of fertilization. So that's a different issue. But anyway, the important point here is, since one ovum is released at each cycle after puberty, supposing a female gets conceived at the age of 35, a female getting conceived, getting pregnant, okay, at the age of 35. So the ovum that was used for the conception is 35 years old. Okay, 35 years old, in the sense it has got arrested in its meiosis for 35 years. And that is the one that is fertilized by sperm. Supposing a female gets pregnant at 40 years, then the ovum is 40 years old. Okay, so in other words, it has been arrested in meiosis for 40 years. Now the problem is, when it is arrested for such a long time in meiosis, during the first phase of meiosis, we know there is phase 1 and phase 2 of meiosis. During first phase of meiosis, when the homologous pair of chromosomes, you remember the homologous pair of chromosomes? And this homologous pair of chromosomes has to go, they have to get separated out and go into two cells. That does not happen. So that is called non-disjunction of chromosomes. Non-disjunction of chromosomes. That non-disjunction of chromosomes actually produce an ovum with one chromosome more, because both chromosomes will go to one side. So ultimately, you get trisomy. Trisomy means instead of 2 chromosomes, you will get 3 chromosomes. You get 23 pairs of chromosomes. So one chromosome would not be 2 pairs, but it would be 3. That is called trisomy. So this happens in certain chromosomes, not in all chromosomes. Now, chromosome number 21, 18, and 30 are very famous for this non-disjunction if the ova are too old, if the female gets pregnant very old. So this 21 trisomy, I think you know it, it's Downs. Okay, so when you get trisomy of 21, you get Downs syndrome. You must have seen Downs children. They are very sweet children anyway. So you get Downs. Then others also, you can check as homework. Trisomy, you just type on the internet, trisomy of 13, 18, 21. So for this, you get Edwards. For this, you get a different name. I don't remember the name. Okay. So you can check this in the internet. Okay. So different chromosomal abnormalities will give different points. So just remember this point. Then in contrast to this, when it comes to the formation of sperms, now here you had only one process, and here you have two processes. Initially, you get spermatogenesis, and then you get spermiogenesis. Now, what happens here is, now this spermatogonia, the male stem cells which are in the testicles, they undergo spermatogenesis and produce this. These are haploid. Okay, haploid spermatids, which are not very good in its mobility. Okay, not that they are immotile, but they are not good when it comes to their mobility. So a spermatid, when they become a sperm, they undergo this process, spermiogenesis, where the sperms acquire their full motility. Motility means ability to move. Okay, because they have to swim to meet the ovum. So then there are two processes, spermatogenesis and spermiogenesis. But even though there are two processes, it only takes 2 months. So sperms, unlike ova, are freshly produced. So even if you take a male with a male at 60 years, the sperms are not 60 years old. Okay. So they are freshly produced. Okay, so that's an important point to remember here. Then in females, there are cyclical changes that are taking place every month. Okay, it's a change taking place in the body. So there are 3 cycles. You label 3 cycles. There is a hormonal cycle taking place at the level of the hypothalamus, the anterior pituitary, and the ovary level. These are the endocrine organs that produce the hormones necessary for the changes in the female. Then there's a uterine cycle taking place physically taking place at the level of the endometrium of the uterus, inner layer of the uterus. Then the third one is the ovarian cycle, physical changes taking place at the level of the ovary. Physical changes taking place at the level of the ovary. Now, these 3 cycles take place from puberty to menopause. Menopause is the cessation of menstrual periods. Puberty is the beginning of the menstrual periods. You call it menarche. The first menstrual period is called menarche. Okay. So from menarche to menopause, or from puberty to, you can call it in simple terms, from puberty to menopause, you get these 3 cycles taking place in the body of a female until there is fertilization and implantation. So at one time point, when the female, the ovum, any of the ova that are released gets fertilized, and the fertilized ovum gets implanted, then that will lead to a pregnancy. Okay, that is conceiving. Okay. Now, when there is a pregnancy, there is temporary interruption of these 3 cycles. Temporary interruption of these 3 cycles for, you can say, roughly 40 weeks, 38 to 40 weeks. Then at the end of, at the time of delivery, still it is not released. Even after delivery, sometimes it takes 6 months for the cycles to come back. 6 months for the cycles to come back. But this can vary. In certain people, it can be very early. In certain people, it can be even later than 6 months. Okay, so they don't get their periods and other cycles back. Okay, so the menstrual periods directly related to the uterine part of the cycle, but that is not alone. It's always linked to the other 2. Okay, a few things about the hormonal cycle. Now, from the hypothalamus, hormones called gonadotrophin releasing hormones are released. And in response to that, from the anterior pituitary, 2 gonadotrophins are released. One is called LH, luteinizing hormone, and the other one is called FSH, follicular stimulating hormone, luteinizing hormone and follicular stimulating hormone. Then if you draw a graph of the level of these hormones, the follicular stimulating hormone, FSH, and LH in the blood of the lady, this female, you will see a graph like this. If you start with day 0, always this day 0 means the beginning, the commencement of the menstrual period. Commencement of the menstrual period, not the end of it. Okay. Beginning of the menstrual period is taken as 0. Then this is roughly, day 14 is roughly the time of ovulation, release of the ovum. And this is the time, if you can put a slash and put 0 here if you want. So that is the beginning of the next menstrual period. Okay, then end of the current, the cycle. So what happens is, from day 0, the FSH and LH levels gradually increase up to day 14. At day 14, there is an LH surge. You call it LH surge. Surge means a rapid increase of LH compared to FSH. So that is actually the stimulus for the ovary to release the ovum. Okay, that is the time of ovulation. Then after that, again, they gradually go down until menstrual period happens. So the reason for menstruation, that is breakdown of the endometrium of the uterus, is actually this lowering of LH and FSH levels, because they come down, especially LH comes down rapidly. After 2 weeks, the endometrium cannot be maintained any further it will break down. I'll go into details of that a little bit later. Okay. Then, if you look at the other part, the ovarian part of it, this is what happens at the ovary level. FSH. Now, I am referring to these two phases here: first 14 days and second 14 days. During the first 14 days, FSH has the main effect. So what it does is it acts on what are called follicles inside the ovaries. Now you have the ovary, large one like this. Inside this, that there are several follicles. Okay. So under the influence of FSH, one follicle, one of these small follicles will develop and becomes a large follicle. Okay. We call it Graafian follicle. And inside that you get the single cell, this ovum. Ovum is inside that. So that is the one that is released and gets fertilized with the sperm. So here, what is shown here are these follicles here. Okay, these are follicles. Okay, so ovary is the big one that you don't see here. Okay, so inside the follicle, as I explained here, you see the ovum. So this is the ovum inside the follicle. Okay, now this development of the follicle in this direction takes place due to the influence of the FSH. Then at the time of around 14 days, ovulation takes place due to this LH surge. Okay. Then after ovulation, the remaining part of the follicle, this part is here. Now the ovum is not there, it has been released. That is called corpus luteum. That is called corpus luteum. It is mainly under the influence of LH now. Corpus luteum. Okay. And then so this is day 0, 14, and 28. Here, under the influence of FSH, now these cells outside the ovum, in the follicle, which are called theca cells, they produce estrogen. So in the first 14 days, during the first 14 days, estrogen level is high. Then under the influence of LH, after day 14 to 28, the corpus luteum will produce more progesterone than estrogen. So the progesterone level is high during the second phase, second 2 weeks of the cycle. So remember this point, that is important for the rest of the discussion. Okay, now if you look at an ovary, this is how you see it. So this whole thing, the big thing is the ovary. Inside the ovary, as I said before, you can see follicles. It starts as a primary follicle, then early secondary follicle, then maturing follicle and mature follicle. Okay, so you don't have to remember any of these names. Okay, they are not important. You just understand what is happening inside. Okay, now inside the follicle, you see this single cell, which is the ovum. Okay, so this is the ovum and this is the ovum here. So this is the one that is released out, ovum. Follicle remains inside and it forms the corpus luteum. Okay, that is the corpus luteum. So at the beginning, it is under the influence of FSH here. And here actually it is under the influence of, I have not drawn, LH. And here they produce a lot of estrogen. And here this one produces a lot of progesterone. Difficult to write. Okay, progesterone. Okay, so this is the one that is released and gets fertilized with the sperm. Now this is actually a fallopian tube. The uterus must have been here, large uterus. Okay, and this is the ovary. Now this ovum that is released around day 14 is taken up by the fimbrial action of the fallopian tube, and immediately after it is taken up, in the ampullary region, this is usually the region where it gets fertilized. You get sperms entering through the vagina. Okay, entering through the vagina and coming all the way, millions and millions of sperms coming all the way through the fallopian tube, and at the ampullary region, usually at the ampullary region, it fertilizes the ovum. Okay, fertilization takes place at the ampullary region. Then immediately at the time of fertilization, it starts division. Okay, mitotic division, mitotic, that is mitosis after fertilization. Okay, so at the time of fertilization, the ovum completes its second meiotic division and becomes mature. and then it starts mitosis immediately after fertilization. So you produce 2 cells, 4 cells like that, while it is moving towards the uterus. I think it's clear to you. Okay, now if you put the 3 cycles together now, hormonal cycle, ovarian cycle, and uterine cycle, this is how you see it. Now you get, these things we have discussed, gonadotrophins, FSH and LH. So then they have this influence, FSH during the first 14 days, LH mainly during the second 14 days. And this is how the changes taking place at the ovary, how the follicles are developing and ovulation takes place here. And then the follicle becomes corpus luteum. Then this is the uterine cycle that we did not discuss so far. Now in the uterine cycle, this is day 0 here, as you can see here. So this is the time when the menstruation starts. So you take it as day 0. So it breaks down for about 4 days. It is different in different females, but in average, it's about 4 days. So at the end of 4 days, the breaking down has been over. Then the endometrium of the uterus starts building up. So you can directly match the diagram above with the diagram below. So when these changes are taking place, at the ovary level, these changes are taking place at the endometrial level. Now, what is important to know here is that you can see one layer in the endometrium, this layer here, you can see it faintly here, that is called basal layer, that is intact. Even during menstruation, as you can see here, basal layer is maintained here. Okay. The other two layers, you can see compact layer and spongy layer, other 2 layers, they break down. They break down during the menstrual period. And again, at the end of the menstrual period, again, they develop back. So every month, compact layer and spongy layer develop and break down during menstruation. Basal layer continues as it is. Okay. Okay. So what happens during the development of the endometrium here is that, let me take this little bit down. Okay. What happens here is that if you see here, now you can see this proliferative, I cannot touch even lower down. Okay. Proliferative phase and secretory phase. Now what happens is under the influence of FSH, during the first 14 days here, we discussed that it produces estrogen. Under the influence of estrogen, this the endometrium of the uterus, there is proliferation, multiplication of blood vessels, there is multiplication of glands and all that. Then the second 14 days, under the influence of progesterone, there is the glands start secreting. So that is called secretory phase. I think you can see here, proliferative phase and secretory phase, 2 phases. Then if there is no fertilization of the ovum, if this ovum is not fertilized with the sperm, which is the usual case, at the end of 28 days, then the uterus, the endometrium starts breaking down, which is called the menstrual phase. So you have proliferative phase, then you have secretory phase and menstrual phase. Actually, this menstrual phase, if you compare with this one, these 4 days will overlap with this part. Okay. So while there is menstruation, a follicle starts developing. Okay. That's why, when you add 14 and 14 is 28 days. Okay. So you can't add 4 days to that and make it 32 days. Okay. So 4 days are within the first 14 days here. Okay. In that area. Okay. So you can match this with this graph, LH-FSH graph. Okay. So you know this now. Okay. So I think I have it here. You have the estrogen and progesterone phases here. The first 14 days, estrogen phase, which is the proliferative phase. Second 14 days, progesterone phase, which is the secretory phase. Now, pregnancy. Now, usually the case is, now this is 28 days. Usually the case is the uterus, the endometrium breaks down at 28 days. Now, if there is fertilization, now if there is an ovum released at day 14, look at this, you can see it. If a sperm comes, fertilization takes place around that area. Okay, so it can be, 1 day before, 2 days after, like, okay, if it is day 14, because of the lifespan of the ovum and the sperm. So around this area, you get the fertilization of the ovum with the sperm. If there is fertilization, it would take another 7 days, like another week, for the fertilized ovum to get implanted. Okay, it gets implanted on the endometrium. Then by the time we reach the 28 days, which is another 7 days, like, there is enough time for some placental tissue. You can see here, some placental tissue has developed, which produces a hormone called HCG. HCG, human chorionic gonadotrophy. So it's a gonadotrophy. It can do the function of FSH and LH. And what it can do is actually HCG, human chorionic gonadotrophy, in the absence of LH and FSH towards day 28, HCG goes and acts on the corpus luteum. HCG acts on the corpus luteum and maintains estrogen and progesterone levels, so that the endometrium does not break down at day 28. Okay, so the endometrium develops further under the influence of estrogen and progesterone produced by the corpus luteum, and therefore the corpus luteum is now called corpus luteum of pregnancy. Corpus luteum of pregnancy, and the corpus luteum operates under the influence of HCG rather than FSH and LH. Okay, so this process mutually help each other. Okay, if there is no fertilization and implantation, this will break down at day 28 because there is no estrogen and progesterone produced by the corpus luteum due to lack of FSH and LH. So this is a nice diagram to show you fertilization. Even though there are millions of sperms coming in, only one sperm is able to penetrate the thick outer wall of the ovum and send its nuclear material inside. So with this, the ovum completes its second meiotic division and becomes a fully matured ovum. And then the mitotic division, mitosis, also starts there. Okay. So this has few diagrams to show you what happens. So you get this completion of the second meiotic division, then you get this, the first mitotic division starting and giving rise to two cells with 46 chromosomes now in 23 pairs, 2N DNA states. So this normal 46 and 2N DNA state is restored here, and ultimately the two cells that are produced are the initial two somatic cells, which are called, together you call the whole thing, the structure, you call it zygote. You call it zygote, it's the two-cell stage. And this term cleavage, it's not a difficult term. It means multiplication. Okay, cleavage means multiplication of cells. So this two-cell stage, zygote stage, it multiplies and produces a four-cell stage. Then it further multiplies, produce 8 cells like that. Then when it reaches around 16 cells, you call it morula. You call it morula. Now the importance here is, if for some reason this gets divided at 4-cell stage or 8-cell stage or even at morula stage, you get completely new embryo. You get a completely new embryo produced by the two cell clumps that are separated out. Okay, even after this you can get, but that's a different discussion. Okay, so this is called cleavage, multiplication of cells in mitosis through mitosis. Okay, then what happens is this morula, this cell ball changes. So I'll draw it here because this is a diagram. So if this is the morula, all filled with cells inside, this further enlarges and forms, I'll get a different color. It produces a cell clump here, and so this outer cell layer remains as it is, the red color one. Then you get the inner cell layer inside. So this is called inner cell mass. You can read it here, inner cell mass. And this is called outer cell mass. Inner cell mass is also called embryoblast because that contributes to form the embryo. And the outer cell mass, this is outer cell mass, is also called trophoblast because that will ultimately give rise to the placenta. Okay. So inner cell mass will give rise to the embryo later, called embryoblast, and the outer cell mass gives rise to the placenta, which is called trophoblast. Okay. It is the trophoblast cells that has the ability to eat into the endometrium. This is the endometrium of the uterus. So you can see it eats into the endometrium of the uterus, and that is what we call implantation. That is what we call implantation. If you draw the uterus like this, it is the upper posterior quadrant behind. Posteriorly, only you get the implantation. That is the best place for implantation of the fertilized ovum. Now this process, now we said ovum is released here and it enters the fallopian tube at the ampullary region, gets fertilized, then it starts cleavage, dividing into cells. By the time this implantation takes place, it is around 1 week has passed. Okay. Now this 1 week is the age of the embryo now, because after fertilization, it's about 1 week. Okay. So around 1 week time, it gets implanted, as you can see here. Okay. Okay, few things about this, the maintenance of the endometrium rather than breaking down like in menstruation. As you can see here, the diagram represents, this is a follicle with the ovum, and this ovum will be released and the follicle will become a corpus luteum. So initially it was under the influence of FSH and LH. Then under the influence of FSH and LH, it produces estrogen and progesterone, which maintain the endometrium of the uterus here. Now, when there is implantation after fertilization, a placenta starts forming, which produces HCG, human chorionic gonadotrophin, which takes up the duty of the FSH and LH anyway, which are going down at 28 days. By the time of 28 days, HCG will act on the corpus luteum here now, and then estrogen and progesterone production continues for another 3 months like this. Okay, another 3 months time like this. Then by that time, at the end of 3 months, the placenta has developed a lot, not small like this. And now by that time, the placenta starts producing its own progesterone and estrogen by that time. Okay. When the placenta produces its own progesterone and estrogen, it does not need the corpus luteum of pregnancy to do that. So anyway, HCG goes down by that time, and corpus luteum of pregnancy degenerates. Inside the ovary, if you check here, you see the degenerated corpus luteum of pregnancy. So you continue with the estrogen and progesterone produced by the placenta itself to maintain the endometrium until the baby is delivered. Okay, now this is a very nice diagram I got from internet. This you can learn a lot of terms and processes by looking at this. I will try to explain it from the beginning. Now you might have difficulty in understanding some of these concepts, but try to understand it well. In case you cannot understand some of the things that I mentioned here, I will arrange another session and play this recording for you all. Okay, just like another lecture, but I will play this same recording so that you can follow it again if you want. Okay, it all depends on you. It's your time. Okay, it's not my time there. Then if you look at this diagram carefully from one end, you can see this says onset of last menstrual period. Onset of last menstrual period. So that is actually day 0. You know this now? Day 0. Day 0. Okay, so focus on that first. So everything is calculated based on that actually. Then from that, if you put 2 weeks, that is 14 days, that is roughly the time of ovulation. If you go by 28-day cycle, okay, that is roughly the time of ovulation. And within 1 or 2 days, it gets fertilized. So fertilization also falls there. So if that is the time of fertilization, okay, if you take from that tip, upper end, okay, that is day 0 of the baby. Okay, that is the beginning of the baby's life at the time of fertilization. Okay, so if you calculate from that day zero, now there are two zeros. This is the beginning of menstruation. This is the time of fertilization. Okay, if you count from that day zero, when it comes to about 38 weeks, 38 weeks, that is the time of the delivery. Okay, delivery. That is the time of the delivery, time the baby is born. That is about 38 weeks. Okay, that is the birth of the baby. Okay, that must be clear to you all. Then if you go to the lower part of the diagram, you can see, now if you start with the time of the first menstrual, the onset of the last menstrual period that the woman remembers, if you calculate from that, instead of 38 weeks, you get 40 weeks. Why? Because of this additional 2 weeks. Okay, that is 2 weeks less than the baby's age. Okay, 2 weeks more than the baby's age, because we have added these 2 weeks. Okay, now the issue is this. Now when a lady, a pregnant lady, comes to the obstetrician, to the doctor, and doctor cannot ask, when did you have your last ovulation? Okay, because the females don't know the time of ovulation. Okay, it's arbitrary. So the only thing the female remembers is that she had a menstrual period. Now the second menstrual period did not come at the right time. Okay, so what she remembers is the onset of the last menstrual period. Onset of the last menstrual period. So what the doctor does is that doctor writes down that date, onset of the last menstrual period, and subtract that date from the current date, today's date. So you get this period of amenorrhea. Amenorrhea is absence of menstruation, because now there is fertilization, there is no breakdown of the endometrium, so there are no periods. So this is called period of amenorrhea, absence of menstruation. Okay, since the doctor calculates this one, you go by this 40 weeks. Okay, that is the time the baby is born. Okay, so that is period of amenorrhea. And if you go by age, this is actually the menstrual age of the baby or whatever. That is the menstrual age. So this is fertilization age. Okay, so remember the difference between the two. So there is a very good anatomical and physiological reason for that. One is fertilization age, other one is menstrual age. When we talk about the embryology, development of the embryo, we go by the fertilization age. We use a different term. We call it, intrauterine life. Okay. So if it is the end of second week, you call it second week of intrauterine life. Intrauterine means inside the uterus life. That is the fertilization age actually. Okay. Then there's another point. Up to first 8 weeks after the fertilization, that is from that side, up to first 8 weeks, this is called embryo. Okay, up to this it is called embryo. Then beyond that it is called fetus. Okay, so people call the whole thing fetus sometimes, but if someone says specifically embryo, that means the first 8 weeks. Okay, then at the till the time of delivery, it is called the fetus. Okay, both terms can be used. Then there's another one. If you take the time of birth, from birth up to 1 year, the baby is called an infant. Okay. First 4 weeks after birth, you can give a special name. You can call it neonate. First 4 weeks after delivery. So neonatal period falls into the infant period. First month of infancy is called neonate period. Okay. So just know these terms. And so this neonatal period, you have neonatal units and all that. Okay. That is for the first 4 weeks. Hope you got something out of this. Then there is another. This is a very nice diagram, I said. No? So there are many things that you can learn from it. So there are other things you can see here. You see this first trimester, second trimester, and third trimester. Now, first trimester means 3 months. Okay. So you have 3 months here, 3 months here, 3 months here. Altogether, 9 months. Okay. Altogether 9 months. Now, going back here again, now this embryonic period, okay, first 8 weeks, even the total 12 weeks, if you take the first trimester, that is the period where most of the organs in the body starts developing. Cardiovascular system, respiratory system, nervous system, all the systems. Okay. After first 3 months, that is here, all systems have developed, starts developing. Now, because of that, this first trimester is a very serious period. So a female should know that she is pregnant during this period and be very careful with eating certain food and especially drugs, taking drugs, because certain drugs can damage the developing organs during this period. Because the most vulnerable period for a baby is during this first 3 months, when the organs are formed. Certain drugs can deform or create abnormalities when the organs are formed. So you will get babies with without limbs, without part of the head, and so on. Okay, so then you should avoid drugs during the first trimester. And even the injections, tetanus toxoid injections are given to mothers, 2 injections per each pregnancy. Even these injections are given during this second and third trimesters. Okay, nothing is given during the first trimester unless there is a serious illness which warrants treatment. Okay, then the same thing, duration of pregnancy and period of amenorrhea. So period of amenorrhea is starting from the beginning of the last menstrual period, and the fetal age starts from the time of the fertilization, which is very close to time of ovulation. Okay, same thing. Okay. Then the early embryogenesis, the part that I wanted to describe in actual, embryology part. Now, early embryogenesis, it's like this. Now, this we have already discussed. Now, this morula, the cell ball with 16 cells, produced what is called blastocyst, which is here. Okay, which has an inner cell mass shown in blue color and an outer cell mass shown in red color here. So you get the development of the embryo and the placenta, as we said before. Okay. Now, with further development, what happens is, now this in the inner cell mass, as you can see here, a cavity develops in the inner cell mass. That cavity is called amniotic cavity. So that amniotic cavity is seen immediately before the delivery also. You see the amniotic cavity filled with amniotic fluid. So that surrounds the baby. How it surrounds the baby, we will discuss. Then by this time, when these cells get separated out and form a cavity, if you look from this side, now this is your eye. If this is your eye with eyelashes, if you look from that side, you will see this, the cells at the bottom will be seen like this, something like this. Okay, if you look from above. So this is like a plate. Okay, that is called embryonic plate, because now this is the one that is going to give rise to the embryo in future. Therefore, it is called embryonic plate. Okay, now still this is within the first week of development. Okay, this is the first week of development. Then there's a cavity below that, which is called the blastocyst cavity, Okay, then after another week, this single cell layer that I showed you, the plate-like thing, has multiplied now. The single cell layer now, like this. Shall I take blue color? It's better, no? So this single cell layer that was there now has multiplied and produced the second cell layer below that. Actually, there are terms used for that. Upper one is called epiblast, lower one is called hypoblast. You don't have to remember these terms. Okay, don't try to remember these terms. Even I forget them all the time. Okay, don't try to remember them. So then, now this is actually two cell layer stage. Two cell layer means actually, now all our body's tissues and organs, they develop from the 3 germ layers. 3 germ layers are the ectoderm, mesoderm, and the endoderm. Ectoderm, mesoderm, and the endoderm. Okay. Now here, the 3 germ layers are not produced yet. Okay. At 2 cell stage, you have only 2 layers. You don't call them ectoderm or endoderm or whatever. Okay. It's epiblast and hypoblast if you want to call them. But it's only 2 layers that are there at the end of 2 weeks. So it's easy to remember. At the end of 2 weeks, you get bilaminar disc. This is called bilaminar disc. At the end of 2 weeks, you get 2 cell layers, bilaminar disc. Okay. Then this further develops. Now there are changes taking place. I don't know whether you need to remember all this. From the lower layer, another cell layer develops and a cavity is formed, which is called exocoelomic cavity. I don't think you don't have to go behind these things, but I'll show you. So this is the 2-layer stage bilaminar disc from your Langman embryology book. And what happens is, compared to this one, you get a layer, mesodermal layer developing between the placental layer, the trophoblast, and the embryo and the yolk sac inside. You get a green layer developing in between. You can see here. Okay, so this side you get the placenta. Inside you get the area of the embryo and the yolk sac here. So this one rapidly develops, and this mesoderm, which is, it's not the mesoderm of the embryo, it is called extra embryonic mesoderm outside the embryo. Okay, that splits and forms a cavity. Now why I said this, so I could have skipped this very easily, but I kept this. The reason is that this is the cavity that surrounds the whole fetus, and it's coverings, inside coverings, and later it forms the chorionic cavity. That's why I showed you this. It forms the chorionic cavity, and this attachment, this one disappears. Okay, and you get only one attachment, which actually, I'll remove this for a moment, which forms one stalk here, which actually forms the umbilical cord later on. Okay. Okay, now this is the plate, this is the area that gives rise to the embryo. Okay, then this term gastrulation. Gastrulation is looks like a big term, but it just means that it is the formation of the 3 germ layers ultimately. Okay, so we were waiting for this. It is the formation of the 3 germ layers. 3 germ layers: ectoderm, mesoderm, and endoderm. What happens is, if you take the 2 layers, we said there are 2 layers, epiblast above and the hypoblast below. Cells from the epiblast migrates in between the 2 layers. Through what is called a primitive streak. You don't have to remember the name again. So it's an opening. Okay, it's a slit-like opening in the upper layer, the epiblast. So cells from here will migrate in through this to lie between the 2 layers. So this is the lower layer, this is the upper layer. So then they produce a 3rd layer between the epiblast and the hypoblast. So that actually gives rise to the mesoderm. So when the mesoderm is formed, then you get the ectoderm outside form. Now epiblast is called ectoderm, hypoblast is called endoderm, and the middle one that is formed is called mesoderm. It is actually the embryonic mesoderm. The previously outside one was extra embryonic mesoderm. You remember? Okay, so this is embryonic mesoderm. So that is called gastrulation. You can forget that term. Formation of 3 germ layers. That happens at the 3rd week. Okay, so this happens at the 3rd week. So you get 3 germ layers: ectoderm, mesoderm, and the endoderm belonging to the embryo. 3rd week, trilaminar disc. Okay. Then now this 3rd week is important because as early as 3rd week, immediately after the formation of the 3 germ layers, the nervous system development starts. Okay, nervous system development starts. The first step in that is, now you have only 3 germ layers. Now at the back posterior side, the dorsal side, the posterior side, the ectoderm changes and becomes a different layer, which is called neuroectoderm. Neuroectoderm. So this, from the neuroectoderm, there is an invagulation develops, okay, and forms a tube, which is called the neural tube. So this neural tube is the one that gives rise to your central nervous system. brain and the spinal cord. We will not discuss that here. That's systemic embryology. We will discuss one day, if we happen to discuss about the brain and stuff. Okay. So why I mentioned this here is that this happens immediately after the formation of 3 germ layers. Okay. So this is how the neural tube is formed. Then this is an important point again. Now all this time, embryo was like a plate here. So it was like 3 layers, like a plate, like a A4 sheet. The only thing is it's not like, it's not square. It's like this. Okay. So it's a plate. Now what happens is this plate starts folding. It bends. Okay. Now if the sheet is like this, if this is the head end, if this is the tail end, then if you have the 2 sides on either side, it folds lateral. These are called lateral folds, and head end folds, and the tail end also folds. What happens is the ectoderm that was lying at the back here, because of this folding, comes to cover the whole thing. So the endoderm inside forms a tube like this again and forms the gut. Ectoderm covers all around, and middle one lies in between, mesoderm. Okay, so that's what happens here. Okay, so this is the embryo with the ectoderm here. This is the endoderm developing, the gut developing from the endoderm, and this is your amniotic cavity, which was above here, amniotic cavity. Because of this folding, it comes to cover the whole fetus, and all around you get the amniotic cavity. Outside this, if you check, you get the chorionic cavity, which I mentioned you in the extra embryonic mesoderm cavity. Okay, I don't know whether you grasp this very well. Now, if you look at this early stage of pregnancy, you can see the embryo inside covered totally with the amniotic cavity. Outside the amniotic cavity, you get the chorionic cavity. Okay, and this is the endometrium, part of the endometrium covering the fetus, and here is the uterine cavity of the uterus. Now, with further development, what happens is this uterine cavity disappears with the chorionic cavity because of the pressure inside. So ultimately, the whole uterus is filled with the amniotic cavity and the baby. This is amniotic cavity. Only in one place there can be little bit of uterine cavity left above the cervix. This is the cervix. Okay, this is the cervix. So if someone wants to do certain investigations to check the genetics of the baby, one can actually send a needle under strict guidelines and take some fluid from the amniotic cavity. You can take amniotic fluid because in this amniotic fluid you get a lot of cells. Okay, dropped from the fetus. If you take a cell and you can do all these karyograms and all, and you can do genetic studies, PCR, and find out the genetic makeup. If there are chromosomal or gene abnormalities, you can actually abort this baby. Okay, so you don't have to wait till this stage. You can do it at early stages here. Okay. Okay, so then because of this folding and everything, now you get a baby here with the ectoderm, then with the endoderm here forming the gut, and mesoderm in between. This is all that is mesoderm. Okay, few things, I will tell few things about mesoderm that is important for your future things. Mesoderm has several parts actually. Mesoderm has several parts. Now it's like this. This part of the mesoderm, now this is the neural tube, developing neural tube. Mesoderm in that area on either side of the mesoderm is called paraxial mesoderm. Okay, and the mesoderm in that area where you get this coelomic cavity surrounding the gut, the peritoneal cavity now, is called the lateral plate mesoderm. Okay, and the mesoderm in between that area is called intermediate mesoderm. Okay, paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. Okay, so this is lateral plate mesoderm. Now, this lateral plate mesoderm, again, this one that is here, further divided into two. There's a mesoderm here, which is called the parietal mesoderm. Then there's a mesoderm here, which is called the visceral mesoderm. Okay, parietal and visceral. You can see the two points here, visceral and parietal layers of the lateral plate mesoderm. Now, the visceral one is also called splanchnic. splanchnic mesoderm. Okay. Parietal one is also called the somatic mesoderm. So either term can be used. Okay. So why I talk about these different types of mesoderms is that later when you learn organ systems, it's important to know from which mesoderm it develops sometimes. I don't know, what they teach in some of these outside universities, but we teach our medical students about this because then they understand certain diseases, especially very well. So this is my classification mesoderm. There's paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. That one has either parietal or somatic mesoderm, visceral or splanchnic mesoderm. Okay. Now these somatic and splanchnic things are very often found. Now if you take lung tissue, later when you say lung tissue, now there is a mesoderm that is inside the lung and in the pleura, the pleura covering the lung. So that is actually then from this splanchnic mesoderm, because that is the inside one. Then if you take the body wall here with the ribs and all, okay, so muscles in that area and the pleura here, parietal pleura, develops from the somatic mesoderm. Okay, inside one from the splanchnic, outside one from the somatic mesoderm. Then this branchial mesoderm, last one, branchial mesoderm, we will not discuss it late now, branchial mesoderm. This mesoderm develops in the area of the neck, in your neck, branchial mesoderm. That is important when you discuss the cranial nerves later, and the head and neck area. Okay, not now. Okay, we will just drop it there. I will discuss few things about this, which is interesting to finish the lecture. I told you this lecture will go on beyond 1 hour. Now the paraxial mesoderm, the mesoderm that is on either side of the neural tube, further divides into sclerotome, myotome, and dermatome. Okay. So coming back, we'll come back to it in a moment. Now, before that, another concept. Now, if you look at an earthworm, now if this is an earthworm, you will see very clearly segmentation of the body of an earthworm. So it's in segments, even outside the body, you can recognize it. We also have this segmented appearance. We also have segments in the body. Now, for instance, if you take, if you take the vertebral column, like this, you can clearly see the segmented nature of the vertebral column. And in the thoracic region, you get ribs attached to the vertebral column. Again, you see that the muscles between the ribs, all these are segmented. Even though you don't see it clearly outside, our bodies are also partly segmented. Okay. And inside the spinal cord, if you take the spinal cord, which is inside the vertebral column, if you take the spinal cord out, you might not see segments in the spinal cord, but when it comes to the function of the spinal cord, it functions in spinal cord segments. Okay, so there is again segmented nature. Now, what happens is, during the development of our bodies, early development, the paraxial mesoderm that was either side of the neural tube, paraxial mesoderm, divides into series of segments like this, which are called somites. Series of somites you can see here. Okay, series of somites. And these somites further divide into 3 parts. Now, this is one somite here. It divides into 3 parts: a dermatome, myotome, and a sclerotome. Dermatome, myotome, and a sclerotome. And this sclerotome actually develops into the vertebra that surrounds the spinal cord here, because spinal cord is surrounded by the vertebrae. So each vertebra is formed by the sclerotome of the somite in that area. And each myotome of the somite in that area gives rise to, when the limbs are formed and the body trunks are formed, they give rise to the muscles supplied by this spinal cord segment. And the myotome and the dermatome produces the skin, dermis of the skin, supplied by the nerves originating from this spinal cord. Okay, so this becomes very important later, because when you do the limbs anatomy, you see dermatomal map of limbs, you see myotomes. Now, if you take, I don't know whether you have read biceps muscle. in the arm that has two myotomes, C5 and C6. That means it has developed from two somites, C5 somite and C6 somite, the myotome of these two somites. Therefore, the relevant spinal nerve coming from that spinal segment innervates this muscle, C5 and C6. I don't know whether you graphed it. It's same with the dermatome and the supply of the sensory supply of the skin. Hope you will understand some of this. You can read books extra if you want, and you can ask questions. Okay, so what happens is when a limb is formed, upper limb or lower limb, these things are dragged into the limb, so that you get a dermatome, you get a myotome in the limb. Sclerotome forms the vertebral body and the arch here.